Hello, everyone. This is Mary Kurek with Frontrunners Innovate, and I have a really special guest for you today. This is going to be interesting. I cannot wait to get his background and his story about how he got into this, because it's going to fascinate you too. This is Bradley Ringeisen, and Bradley is the executive director of the Innovative Genomics Institute, and currently is in Berkeley on a stint, he calls it, because he is a Southern guy. We know that, Southern from Virginia in the United States. We are very global, Brad, so I have to tell people that where it is located. But we want to talk about genomics. We want to talk about you first. So talk to us a little bit about what brought you into this interesting line of work.
Yeah, thank you so much, Mary, for having me today. I'm really excited to share some of my experiences and more information about the IGI. But yeah, I've been here for almost a year and a half now at UC Berkeley's Innovative Genomics Institute, which was founded by Jennifer Doudna of world-renowned fame for discovering the CRISPR-Cas gene editing and developing the CRISPR-Cas gene editing toolkit. But I started here back in the summer of 2020. And my last job was actually with the Defense Advanced Research Project Agency. For most of you, you would probably know that as DARPA, which is sort of the innovation hub for the Department of Defense. But prior to that, I was a research scientist and a chemist and a biologist for the Department of Navy at the Naval Research Lab. So my entire professional career, I actually worked for the Department of Defense as a scientist, working on what biology could do for the Department of Defense to help protect soldiers and provide alternative energy and all kinds of interesting things back in my background. Mm-hmm. But when I was at DARPA, I specifically actually led the Biological Technologies Office, or BTO, at DARPA. And I was there for about 3.5 years. And when Jennifer Doudna sort of called and sort of started to ask about whether I would be interested in this position, I really honestly wasn't sure if this was the right jump or not, because I'd been working for the Department of Defense and the government for most of my career. But then as I started to learn more and more about IGI, I started to realize that maybe the opportunities out on the West Coast and out here at University of California, Berkeley, and working with Jennifer actually allow me almost a bigger platform and to actually sort of get outside of the application spaces that biology can impact for the Department of Defense. And I started seeing in my role at DARPA, again, putting 50, 60, $70 million behind really innovative, like cutting edge technologies. We were funding Moderna's RNA, mRNA vaccine way back in 2012, 2013, before it really became big. So you can have a big impact, but it was all about the Department of Defense and what you could do for those applications. And I just kind of started to feel like the second time, the third time, I was thinking about these same applications over and over again. Yeah. And I'm actually really, really passionate about climate research and what biology could potentially do in the field of climate. And Jennifer was on board, and she really wanted to try to push her institute, the IGI, not only towards health and trying to save people's lives with CRISPR therapeutics and CRISPR cures, but also seeing what CRISPR could do for the climate, to see if we could really start to make crops more adaptive to climate, to be able to maybe even mitigate and capture carbon through edited crops and plants and bacteria. So that's really what got me, was that push to not only try to save lives through CRISPR cures and treatments, but also the possibility to really be able to fulfill something that I'm really passionate about, which is trying to help in this fight against climate change as well.
What an interesting span from healthcare to climate, and who knew there would be an intersection there?
It really is. It's where a lot of the commercial interest is, where all the startups and all the capital is all in on health. People understand you can edit human cells, you can cure genetic diseases now. There have been individuals that have literally having no side effects and no sickle cell disease symptoms, as one example. But so people are just pushing money in on the health side. Yeah. But I'm trying to raise awareness that, look, CRISPR can edit plant cells and bacteria just as well as it can human cells. And so there's a tremendous amount of opportunity to be able to capture more carbon and maybe be able to pump more carbon underground. And so it's not actually as different as what you might think it would be.
Interesting. Before we get too far into this, for those people who are listening that may not understand the whole CRISPR concept, can you please give us the short version, the definition of that?
Yeah, so people probably are familiar with the fact that you can sequence the genomes now. You can read the genomic structure and genomic character of a living organism, whether that's a bacteria, whether it's a plant, whether it's a human. You can basically sequence and get all of the letters, the A's, G's, C's, and T's that make up the human genome. But as of 10, 15 years ago, you actually have the ability as well to go in and specifically modify to either knock out and turn off a certain gene or potentially add in new functionalities and add in new genes into the genome. And that's essentially called gene editing. And what Jennifer and her collaborator, I always mispronounce her name, Emilio Charpentier. It's a French name, but they both won the
I'm not the
Nobel
only one
Prize.
that does that. So.
I'm sorry. Back in 2019, won the Nobel Prize in chemistry in 2020 for the discovery of the CRISPR-Cas system of gene editing. And that CRISPR system has two things. It has what's called a guide RNA, which essentially is the heat-seeking missile. It has the ability to be able to lock in, to be able to identify exactly the right part of the genome that you want to be able to edit and to be able to change. And then you have this other system called a Cas protein that then essentially is the scissors that can snip and either knock out a gene or potentially then add in a gene. And so you can now both read the genome and you can also now change and modify the genome, which ultimately gives you new functions. You can cure a disease, you can make a plant resistant to a disease, you potentially could make a plant like a crop like rice or corn operate without the need for fertilizer, potentially. It's just as one specific example. So as soon as you can go in and you can edit and add function, that allows you now to sort of create all of these new products that could potentially have really life-altering implications.
So, okay, more stupid questions.
Okay, there are no stupid
we're
questions.
on this vein. Okay, for those people watching who may have either friends or family or themselves maybe fighting a terrible disease, let's say cancer or something like that. Where are we at in the stage of development with CRISPR and this genomics? And the all the authorities out there, whoever has to bless all of this stuff so that it becomes something that our family physician can deal with, or whatever transplant centers can deal with. How far are we?
Yeah, it's so much easier to answer that question now than it was 2 years ago, because as of 2019. There have been humans that have been positively affected by CRISPR, okay? So it has been in, human cells, and those human cells have been then transplanted into humans. Victoria Gray was the first very, very heroic and very brave individual who was suffering from sickle cell. And she volunteered and signed the consent forms to be able to have her cells, her blood cells, edited by CRISPR. And what they did was essentially turn on another way for her body to make hemoglobin, which is the iron-carrying molecule that's in your blood. And sickle sort of modifies that hemoglobin and doesn't allow it to carry blood nearly effectively enough. So her cells now carry these edits and these changes to be able to actually produce hemoglobin that's functional, and her symptoms have been effectively eliminated. And so that's so as of 2019, that's occurring. Now there's just an avalanche of human studies and that where CRISPR is showing again and again and again to be effective at curing disease. And so now the caveat there is that it's for relatively simple diseases that maybe have one gene that is mutated, that doesn't work properly. And you have the ability to then go in, target that one gene and be able to fix it and modify it. Or if it's a bad gene, then you go in and you can knock out and modify that gene. And so that's where we are right now. It's amazing that it's happened so well. It's been done in liver, it's been done for blood cells, and we have the ability to actually go in and cure those simple genetic diseases. Okay, that's number one. Number two, on the sort of plant side and on the crop side, we can do the same thing. Our institute can edit over 25 different crops, anything from the chocolate cacao to rice to maize and corn and tomatoes. And so we have the ability to be able to do that. And there actually is a CRISPR modified tomato that is now actually available for consumers to purchase as well. So it's such an easier answer now because it's in the real world. It's in humans and it's in plants. And I will make the quick statement too, which is really important to describe, Mary, is that CRISPR modified crops are usually not considered GMOs. This is something that you can actually create where you knock out genes and there is no genetic modification. There's no new genetic material that makes it a GMO that's in there. So it's very, very important for people to understand that almost all of the crops that are modified by CRISPR are not considered GMOs by the Food and Drug Administration or the EPA.
Yeah. So it sounds like from the crop perspective, produce, if you will, that it's moving forward. So there's some things readily available in that space, but from the human health perspective, are we still in clinical trials? Are we?
Yeah, still clinical trials, and they're very targeted. They're very targeted towards specific diseases. But look, this is a stepping stone thing, and we always talk about CRISPR as a platform technology. Yeah. This is something that because you can modify that genetic code, it very easily could target gene A or gene B or gene C. And so it has a knob that you can turn very, very easily to turn it to different diseases. So once we start seeing successes in these clinical trials, Mary, it's only going one direction. It's going and it's just going to blast off exponentially. And I promise you that the same thing's going to happen on the agriculture and climate side as well, because there's just such a need for that to happen. I was at a conference up in Boston 2 weeks ago at Ginkgo Bioworks, and the CEO of Ginkgo, which was, by the way, the largest IPO in biotech history, and the CEO for Ginkgo was stood up on the stage at his conference and basically said, the next 100 unicorns are going to be in climate and ag. For exactly that reason, because there's so much opportunity and so much green pasture that's out there, no pun intended, where you make really big impacts in that space.
The funny thing about climate is I had no real concept of how it can affect or impact so many other things. When you can fix that, you can almost fix just about everything else.
I mean, look, we're talking about crops being susceptible to more disease, higher temperatures, more drought, less rain. Those are things that we're actively working on at the Innovative Genomics Institute.
That's what I wanted to get into. Yeah,
drought tolerant rice. We're looking at ways to modify crops to be able to increase how much carbon that they can capture. And then pump that carbon underground with longer roots and deeper roots and ways to be able to capture that and hold it in the soil for longer periods of time. These are all things that are possible in the next few years, honestly. And it's really is what's really exciting.
Are you case studying or doing any kind of trial with that kind of thing right now? Let's just take, for instance, the rice in the drought.
Yeah, the drought tolerant rice. Absolutely. It's a great question, Mary. We actually are partnering with an organization called CIAT, CIAT, and they're going to take that drought tolerant rice that uses less water, and actually field it in an international field trial in Colombia, in Colombia. And so that's something that's actually going to start next year. And then on the other side, we have a clinical trial in sickle cell that we're going to dose patients next year in 2022. And there's also an immune deficiency genetic disease that we're also expecting to dose patients in 2022 as well. So we've got it going on both sides, where we're doing international field trials of some of these crops. And at the same time, here in the United States, partnering with University of California, San Francisco, the medical school over in San Francisco, we're going to be starting 2 clinical trials next year as well.
Fantastic. I'm thinking, because we see a lot of it on the news, and lately there has been that climate produced situation in the southern part of Madagascar, which I'm very familiar with because I have a relationship with monarchy there. And that's the first time that I think the news reported that was absolutely a result of climate change is the drought that they're experiencing and the famine that's happening that is associated to that. So I think
you also see on the other side, Mary, you see flooding. And so you can actually create flood tolerant rice as well, to be able to balance it on the other side. So you're seeing both, you're going to see more and more and more extremes.
Exactly. There are some places in, the Philippines, my goodness, about every other year, they get smacked with some kind of tsunami that just floods everything. So
I'm a big believer in technology. I'm truly being at DARPA. I have faith that technology can solve some of these problems. And I think that there is an ethical and regulatory pathway to be able to use technology to be able to do this. That's one more thing that I should say, as we're talking about places like Madagascar and the Philippines. Jennifer founded the IGI on the premise that CRISPR-Cas, it can be a technology that can help raise all lives and help really affect those that need it the most, that it does not have to be in the hands of just the elite and those that have the money to be able to pay for things. So most of our focus, we work with these international nonprofit organizations on the agriculture side to be able to provide these seeds and other things at cost. And then at the same time, we're hoping to be able to use technologies to be able to lower the cost on the healthcare side of some of these treatments right now cost $2 and $3 million. And we want to be able to bring that down so that all insurances and Medicare will be able to pay for these things.
That's phenomenal, because whenever something like this new comes out, it is, before Medicare or any other insurance will pick up anything, it's out of pocket and it's outrageously expensive. So it's almost like the sicker you are, the more devastating, the impact is going to be on the wallet.
There are some companies that are actually pulling out of Europe because Europe said, look, we just can't pay for this. And so they're going to focus on the United States market instead of Europe. So it's a real problem. And our institute's really trying to develop technology that can cut out some of the really expensive steps and really just go directly to be able to lower those costs.
Fantastic. So that's kind of that's the picture, I guess, of what's going on now from what you just said, of the things that you're working on. What's in the future? What's in, you already told me 2022, you've got a couple of clinical trial things going on.
That's still right here. I mean, we're going to shoot so much higher, Mary. So that's a great question.
I know. So that's what I'm excited about. So what's coming? What do you see coming that's fantastic?
So let me talk about both sides again. Let me talk a little bit about the ag, and then I'll talk a little bit about the health, and I'll end on some of the health implications because I think it's pretty astounding. Okay. But on the agriculture side, look, agriculture is part of the problem right now. Bill Gates was famously saying that, of all of the emissions from humans, like agriculture is actually up to maybe 25 to 30% of those emissions. And it's just going to get worse as the population of the world gets higher and higher and higher. If we don't change the way we make and produce our food, it's going to be 50% of the emissions that we have in the world. And so we cannot continue on the same path that we're on with respect to how we grow and grow and feed the world right now. Okay. So right now, if I was to shoot really high over the horizon and say, what could CRISPR and gene editing do? I think it really boils down to two things. We are headed towards a different climate. That's just going to happen. All of the UN reports, there's just a report on the front page of the Washington Post this morning, 2.5 degrees. That's what's going to happen. There's going to be more extremes. CRISPR can be used to try to help adapt crops to that changing climate. Okay. Whether that is making it disease resistant. Look, we're working at the banana. The banana is under threat from a soil fungus right now. We might not actually have bananas 5 or 10 years from now, based on the fact that it's spreading across the world. So we are working on that right now. We're working with several countries in South America and Latin America to be able to help fund that work. Other pests like the cacao, which is responsible for chocolate, also is under attack by a virus that could really threaten the world's supply of chocolate. And then things like wheat, right, is also under attack by different pathogens. So I really think that we can try to adapt these crops to be able to help keep continuing to feed the world, even in this very, very changing climate, number one. But then number two, I think we can actually help solve climate with plants and CRISPR, helping soak up more carbon, helping to be able to store more of that carbon into the soil, helping soil bounce back, helping it be more fertile, where there's more carbon, more nitrogen in the soil. And all of these things I think are possible within the next 10 years. So let's reverse that curve. Let's not keep the carbon footprint and the greenhouse gas emissions associated with growing our food. Let's turn that curve over. And I think we can actually get maybe to a net zero farm. And I think that's something that we're really, really excited about trying to work on in our institute, but then also help it be part of the solution. You hear about these carbon capture engineered systems where they're sucking air and forming, stable carbon products. I think nature can help with that as well. And I think that CRISPR can help nature actually capture more carbon and to be able to store it as well. So to me, that's the future. In 10 years, I want to look back and think that I've done everything that I can to use gene editing to be able to help store more carbon and create a net zero farm. That's really what I would like to do on the agriculture and climate front. If you pivot quickly to the health side, I talked to you about genetic disease. A lot of these diseases have 10 people, 100 people. Sickle cell is the exception, where in the United States, there's 100,000 people. But I would really love to see gene editing be used for more common diseases. Things like neurodegenerative disease, Parkinson's, Alzheimer's, and fighting things like cancer. And we have study groups and we're starting projects inside the Institute right now because we really want to shoot over the horizon and affect these more common diseases that are affecting millions and millions of people, not just tens to hundreds. We're doing both. Those rare diseases and what people call neglected disease, we are absolutely working on those rare and neglected diseases. But we're also thinking about ways to be able to deliver CRISPR to the brain and the central nervous system to be able to potentially affect some of those neurodegenerative diseases. And we're working with our collaborators at UC San Francisco, who are already starting to do some therapies directed towards cancer, but there's still some really big challenges out there for solid tumors and to be able to sort of deliver the therapeutics directly to your body. And we're working with those UC San Francisco partners to be able to do that. So I think the future is very, very bright on both sides, and we're really excited to just make really big impacts in the next 10 years.
This is fantastic stuff. What comes to mind is before the pandemic hit, I had done a string of special edition articles on diabetes. And so I know that's like an epidemic everywhere. Is there any hope for those folks?
Diabetes is one of those difficult things. I haven't personally thought about that very much. That's one of those things that I think that there might be solutions where you go in and you modify and turn on some of those mechanisms to be able to form insulin or things inside your body. I think that there is an opportunity there, but you also, that's one of those systems that has to be very, very regulated inside your body.
There's a lot of things that tie into that. And I think, there really are. There's a whole lot of things that tie into that.
So that's one of those challenges, is that it's not just turning on and have something produce something all the time. You have to have a give and take where it has to be regulated and modified. Sure. I don't think it's out of the question, but that would be the challenge in that particular problem.
Impacting millions, worldwide for sure. So, my last question, as I mentioned to you, is who do you need? What type of people do the Institute, or does the Institute need to help move things forward? Partners, clinical trial, or other corporations that are helping to fund trials, or tell me a little.
That's really it. We need partners, Mary. We really need partners. And about 80% of our institute is run philanthropically. So this is something where Jennifer and I are in a big fundraising campaign right now to support the institute. And we have these big visions on both the health side and on the ag and climate. And we're really looking for partners to be able to join us, to be able to help bring those visions to reality. And who better to partner with than one of the most recent Nobel Prize winning scientists in the world, an ex-DARPA guy that just wants to help lower healthcare prices and save the world from climate disaster. So why not join us? I think there's really real opportunities there. But there are corporate partnerships that I think actually can work with us as well. We generate a tremendous amount of intellectual property in the Institute. Anywhere between 15 to 20% of the intellectual property for the entire campus of UC Berkeley. So there's a lot of opportunities for us to partner with startup companies, to be able to partner with venture capital companies. We're starting a new project now where the venture capital company gets access to the intellectual property that's generated. They fund us to do innovative research, And then they're able to get access to the startup companies that we form, so that they have the ability to be able to fund those companies into the future. So there's a tremendous amount of opportunity, both on the philanthropic side, as well as on the more commercial and venture side, to be able to partner with us.
Fantastic. If somebody were interested in doing so, would they reach out through your website?
Yeah, you can go right to our website. It's innovativegenomics.org, I believe. I don't go, yeah, innovativegenomics.org.
We'll get it from you. We'll put it in with this. So yeah,
Perfect.
that's fine. So other than that, any other volunteers, resources, anything else, or is it?
Well, we always need good scientists, Mary. And so if you want to go to graduate school, come to UC Berkeley for graduate school. If you're looking to do a postdoc, come to the Innovative Genomics to do a postdoc. That's the engine, that students are the engine that drive innovation. I've been in this business long enough to know that. So we absolutely would want undergraduates, graduate students, postdocs, anybody that wants to join. Jennifer, join UC Berkeley. It's a great place. I'm out here at Berkeley right now. The weather's great. The science is great. I don't think I could think of a better place to go to graduate school or to do really amazing science.
Fantastic. Well, all of you watching, you now have your marching orders for sure. This is such great stuff, and we just wish you all the best and very well as you move forward. It's almost like you're part think tank, part R&D, and
it's an exciting place to be.
Just definitely moving the needle on change that needs to happen. So always, disruptive sounds like such a negative word. So I'm just going to say you're making positive impact. So we're just going to go with it that way.
Thank you so much.
Yeah, thank you so much, Brad, for joining me for this conversation. It's been enlightening, to say the least, and we'll do our best to kind of move things forward. If you'll stay with me for just a second, I'm going to say goodbye to everybody else. If you're watching this on YouTube, go to www.frontrunnersinnovate.com, where you'll have the full interview. You'll have, hopefully we can get a bio from Brad, because I know he probably does some speaking or what have you, and some links so that you can follow along and check in on the progress and connect with them if you want to partner, or if you'd like to connect with them to do an internship or whatever you'd like to do. So we're going to leave it with that. So bye, everybody. And thank you, Brad. And just hang with me for just a second.
Sure. Thank you so much.
Bye-bye.